Basic concepts in embryology. Embryology, the study of early organism development, is grounded in several core principles that have shaped our understanding of how organisms grow and form from a single fertilized cell to a complex organism. These foundational concepts include epigenesis, preformation, and embryogenesis. Below, I will provide detailed information on each of these concepts. First, epigenesis. Definition: epigenesis refers to the idea that an organism develops progressively from a simple undifferentiated state to a complex, fully formed organism through successive changes. The key aspect of epigenesis is that the embryo develops through a series of gradual and stepwise processes where the structures of the organism emerge progressively from the fertilized egg. Historical context. The concept of epigenesis was first proposed by Aristotle in ancient Greece. Aristotle argued that organisms develop from an initially undifferentiated state, where the fertilized egg gradually transforms into a complex organism with specialized parts. Epigenesis was a significant shift from earlier beliefs such as preformation, which proposed that organisms were already preformed within the egg or sperm and simply grew to adulthood over time. Key features: development through stages. Epigenesis emphasizes that an embryo forms by the differentiation of cells and tissues through various stages, such as cleavage, gastrulation, and organogenesis. Role of environmental factors. Epigenetic processes in development can be influenced by environmental factors, including nutrition, hormones, and temperature, especially during early development. For example, maternal nutrition during pregnancy can affect fetal development and gene expression. Modern understanding. Today, epigenesis is understood through molecular biology as a process involving gene expression regulated by the epigenome, which controls how genes are turned on or off without altering the DNA sequence. Epigenetic modifications like DNA methylation and histone modification play a crucial role in regulating cell differentiation during embryonic development. Second, preformation. Definition: preformation is the concept that the organism is fully formed within the sperm or egg at the moment of fertilization, just on a much smaller scale. According to preformation, all of the future organism structures exist as miniature versions of themselves called homunculus in the germ cells, sperm or egg, and simply grow larger over time. Historical context. Preformationism dominated early biological thought before the development of modern embryology. Preformation was widely accepted during the 17th and 18th centuries, with notable proponents such as Anton van Leeuwenhoek, who believed that the sperm contained a tiny preformed human being, the homunculus. The theory was eventually replaced by epigenesis and modern embryology, which are supported by the observation of development as a gradual process. Types of preformation. Spermist. Preformation. According to this view, the miniature organism is contained in the sperm, and the egg simply provides nourishment and an environment for growth. This idea was supported by Anton van Leeuwenhoek and some early biologists. Ovis preformation. This theory suggested that the organism is preformed in the egg, and the sperm simply contributed a trigger or vital force to activate development. Marie-François Xavier Bichat and Caspar Friedrich Wolff believed in this theory during the early 19th century. Critique and displacement. The theory of preformation was disproved through the development of modern microscopy and better understanding of cellular biology. Observations of fertilization, cell division, and differentiation demonstrated that development is not based on preformed organisms, but arises from gradual cellular interactions and differentiation. Third, embryogenesis. Definition. Embryogenesis is the process by which a fertilized egg develops into a multicellular organism, which then differentiates into specialized tissues and organs. It encompasses the series of stages an embryo undergoes, starting from the single-cell zygote and continuing through various developmental phases until the organism is fully formed. Key stages in embryogenesis. Fertilization. The sperm and egg unite to form a single cell called the zygote. Fertilization is the first step in embryogenesis, combining the genetic material from both parents. Cleavage. Following fertilization, the zygote begins to divide rapidly through a process called cleavage, forming a cluster of cells without increasing the overall size of the embryo. This forms a morula, a solid ball of cells, and eventually a blastocyst, a hollow sphere of cells. Gastrulation. This stage marks the formation of the three germ layers, ectoderm, mesoderm, and endoderm, which will later differentiate into specific tissues and organs of the body. Ectoderm forms structures such as the skin and nervous system. Mesoderm forms muscles, the circulatory system, and other connective tissues. Endoderm forms the gastrointestinal tract, lungs, and other internal organs. Organogenesis. This is the stage where the three germ layers differentiate into organs and organ systems. For example, the heart, brain, and kidneys begin to form and develop specialized functions. Fetal development. Following organogenesis, the fetus continues to grow and mature, with further specialization of organs and tissues. The fetus prepares for birth, where the organs become fully functional. Molecular mechanisms in embryogenesis. Embryogenesis involves a series of molecular signals that regulate cell behavior. Key signaling pathways such as Notch, WNT, and Hedgehog help to control cell fate, patterning, and differentiation during development. Gene expression regulation is essential during embryogenesis. The early stages of embryonic development are largely controlled by maternal mRNAs and proteins, while the zygote's genome eventually becomes activated, leading to a more complex and autonomous regulation of development. Key events in embryogenesis: cell division and growth. The zygote divides rapidly to produce a blastocyst, followed by a rapid increase in cell number and the establishment of cellular diversity. Cell differentiation. As cells divide, they begin to specialize into different types that will form specific tissues and organs, e.g., skin cells, muscle cells, nerve cells. Morphogenesis. The process through which cells and tissues arrange themselves into distinct shapes and structures. This is crucial for the proper formation of organs and limbs. Modern embryogenesis research, stem cell technology. Embryonic stem cells have the potential to form any type of cell and hold promise for regenerative medicine, allowing scientists to create tissues and even organs. CRISPR-Cas9. Gene editing tools like CRISPR are being used to study embryonic development by knocking out genes and observing their effects on embryogenesis. This can help uncover the functions of specific genes in development. Comparing epigenesis, preformation, and embryogenesis. Concept, description. Key features, epigenesis. The idea that development occurs through gradual differentiation from an undifferentiated state. Embryo develops progressively, influenced by environmental factors. Preformation. The belief that the organism is already preformed in the egg or sperm and simply grows larger. Early versions of the organism exist at conception. Embryogenesis, the actual process by which a fertilized egg develops into a complex organism, includes stages like cleavage, gastrulation, and organogenesis, with cellular differentiation and tissue development. Conclusion. These fundamental concepts, epigenesis, preformation, and embryogenesis, form the foundation of embryology. While preformation has been largely disproven, it played a pivotal role in the history of developmental biology. Epigenesis, which emphasizes the gradual formation of complex organisms, remains central to modern embryology, and embryogenesis provides the framework for understanding the processes that give rise to diverse and functional tissues and organs. Modern research continues to build on these concepts, providing insights into developmental, genetics, stem cell biology, and regenerative medicine.